start the second video on the chapter on enzymes. Now this is biology 9700 and this is the chapter on enzymes, which is the third chapter. Now basically what we want to discuss in this is what are inhibitors. This is one component of the chapter which has not been covered. So if you look at this diagram, this is the enzyme, this is the substrate, and we have something which looks a little like the substrate, but it's not exactly like the substrate. And as it can slow down the reaction, like for instance this inhibitor, if it fits in, well, no product is going to be formed, so it is going to slow down the reaction. Now because it has sort of a shape which is similar to the substrate, it is called a competitive inhibitor. And that which has a shape other than, so if we have something which is not the same shape as, and it fits somewhere else, it can fit to a site other than the active site, and it can change the shape of the active site. So the substrate no longer fits. Now those are called non-competitive inhibitors. So we have two types of inhibitors: competitive and non-competitive inhibitors. Now as we study enzymes, we see a lot of graphs in this chapter, and a lot of MCQs are on the graphs. So on this graph we have substrate concentration on the x-axis, and we can say it's in percentage, or we can use it some other units as well. And we have the rate of enzyme on the y-axis. Now I'm just giving you a very simple example, and I'll say there are 100 active sites. Now if we only have 10 of these, then of course the rate is going to be slow, and then it's going to finish. And say let's increase this to 20. Let's increase this to 30. So the rate keeps on increasing. Now a point comes when we have the same number of substrate molecules. Okay, so the rate keeps on increasing, and now if we increase the substrate even more, the rate becomes constant. The rate becomes constant because now the enzyme has become the limiting factor. The enzyme has become the limiting factor because now the only 100 active sites are available. But even though we are increasing the substrate concentration, we might increase it even further, we might increase it further, but the rate becomes constant. So at times they give you this graph and they say, okay, point X, what is the explanation for point X? The explanation for point X is all active sites occupied. You can say that, all active sites occupied. Or you can say enzyme is the limiting factor. Enzyme is limiting because you see you only have 100 active sites, and if you increase, so is the limiting factor. So point X can be explained in a number of ways. Now even though we've increased the substrate, but that doesn't make any difference. This, we say the rate is constant, and this is called Vmax. So what did I say? Rate is constant. So even though we have increased the substrate, so let's look at the situation again. This is the situation. We only have 10 substrate molecules, and this would be somewhere here, this point of the graph. We increase it to 20, we increase it to 80, and then we say, okay, this is the point where we find that it will be the same number of active sites. So all active sites will be occupied. Now even if we increase the substrate, it's not going to make much of a difference, is that the rate will become constant. Now, in a similar shape of the graph, what we've done is on the x-axis we have time, and on the y-axis we have volume of product. Now, the reaction which we're talking about is catalase, breaks down hydrogen peroxide to oxygen and water. Now, if we are measuring the volume of oxygen produced, if somehow we've got a gas syringe and we are measuring the volume of oxygen produced, we find that the volume increases. But after some time, the volume becomes constant. Now, this is very much like this previous graph. But here, the answer is, if I say what is the answer, what is happening at x? Why is the volume, say the volume is 10. Now, why is the volume of the product remaining 10? The reason is that all the hydrogen peroxide has been used up. So all the substrate has been used up. So a similar graph, but now look at what is the labeling on the x-axis and the y-axis. The x-axis is time, and on the y-axis is the volume of the product. Here the volume of the product is the oxygen in this situation. So if I had to ask you what was happening at x, you would say all the substrate used up. Because the product now, why is it that no more product is being formed? Because all the hydrogen peroxide has been used up. So no more of the substrate is left. So no more substrate, no more oxygen being produced. So a similar graph with the same sort of shape, but here it was substrate concentration and here it was rate of enzyme action. While in this graph is time, and on the y-axis is the volume of the product. So please be very careful whenever you're doing MCQs on the chapter on enzymes. Please always read what is on the x-axis and what is on the y-axis. Now, another topic which needs to be considered is the Michaelis-Menten Constant. Now, Michaelis-Menten Constant is also represented by the letters Km. Now, what we need to understand is that first we need to calculate what is the Vmax. The Vmax is 100. So we have to calculate half the Vmax. Now, half the Vmax is 50. And then you draw a line straight, and then you read off what is the substrate concentration. Now, whatever this value is, that is the Km. So say it is 5. Here we have 2, 3, 5, 6, 7, 8 and things like this. So the substrate concentration, the value of the substrate concentration which you have to study which is going to be the Km. So basically Km will be the substrate concentration. Now if we draw another graph, and it reaches the same Vmax, now you can see how it has totally changed the Km value. The Km value has become now more. Initially the Km value was this, now the Km value is this. So there's a very simple rule. We say the lower the Km value, higher the affinity. Basically, affinity means how much it is reacting, how quickly it is going to react with the substrate. So if we look at this one, which is say A, enzyme A, and then we look at this one, which is enzyme B. So enzyme A has a higher affinity because it has a lower Km. So the lesser the Km value, so when you look at the Km value, this is 5 and this is 7. So the lesser the Km value, the higher the affinity the enzyme has for the substrate. So we study different enzymes, like we study amylase. We study amylase from humans, then we study amylase from maybe another animal, or we study amylase which is present in potatoes, and we get an idea is which enzyme has a higher affinity. So lesser the Km value, higher the affinity. Now this is a very simple thing. So Km is, first we need to calculate the Vmax. So we just extend the line. Then we calculate half the Vmax. And then we see where it dissects the graph, and then what is on the x-axis. So simple rule, lower the Km, higher the affinity. Now if you look at this graph, you can see we have substrate concentration here on the x-axis, and we have rate on the y-axis, rate of enzyme action. Now we normally get a graph in which as we increase the substrate, then the rate becomes constant because the number of active sites, which is 100, becomes the limiting factor. But now look at the situation in which we have added 10 substrate, and we've also added 10 an inhibitor, which is a competitive inhibitor. So the rate is going to slow down. Now when the rate is going to slow down, but slowly, what are we increasing? We are increasing the substrate concentration. We are increasing the substrate concentration. So after some time, we can have, this is 20, then this is 30. And as we keep on increasing this, we come to a very interesting conclusion, is that if this is 100, then these are only 10. They have no chance to fit into the active site. And if they fit into the active site, they do not form a product. So as we keep on increasing this, these poor competitive inhibitors have no chance. So if you look at the graph is going to ultimately go and reach the final end. Now this is a very typical graph for competitive inhibitors. And this is what you have to understand, is that when the inhibitor is less and the substrate is less, then of course there's competition. Like there's 10 of these and 10 of that. Then of course if this inhibitor fits in, no product is formed. But it still delays the action, because if that's fitting in, then we're hoping that this is going to move out and then the substrate is going to fit in. It's just like you've got this, and you've got this, and you've got this, and you've got them in a bag and you're shaking them. So if this fits in, then no product is formed. But if this fits in, then a product is formed. So the rate of reaction is measuring product per unit time. Now this is competitive inhibitors, and this graph is a very typical graph in which, of course, as we increase the substrate concentration, the graph will reach the Vmax. Now the other type of inhibitor is a non-competitive inhibitor, and it fits to a site other than the active site, and it changes the shape of the active site. So that means if I have, say, 10 of these, so 10 active sites no longer functioning, 10 active sites not functioning. So we only have 100 minus 10, 90 active sites working. In non-competitive inhibitor, it fits to a site other than the active site, or sometimes it also fits to the same active site. And it changes the shape of the active site. So we're never going to get a graph which is going to reach the Vmax. So non-competitive inhibitors will attach to the enzyme and will change the shape of the active site. So if the active site shape is changed, then the substrate no longer fits. And so that means out of 100, 10 active sites have changed shape. So the graph will never reach the Vmax, and non-competitive inhibitors sometimes are reversible, because if they are removed, then the active site will again regain its shape. So this can also happen, and then of course the shape will be regained. And this is sometimes important. Why? Because we have to slow down certain reactions at times. So non-competitive inhibitors attach to a site other than the active site, and they can change the shape of the active site. So if the shape of the active site is changed, substrate will no longer fit. Now, maltose is a sugar which is very much needed in the candy industry, chocolates, drinks. So it's used in the beverages, and we need maltose. Now, starch we can get from a lot of stuff, and it's very cheap to get it. You can get it from uneaten bread or old remaining rotis and old potatoes, or even very bad quality rice. You can just boil it and you can get a starch solution. Now to this starch solution, if you add amylase and you keep the temperature right, after some time all this starch will have been converted to maltose, and all the starch will have finished. So starch is all finished, and we have now maltose. We have maltose and we have the amylase. Now for this to separate it, because we only want the maltose, we don't want the amylase. So the amylase is not required. You will need a molecular filter to separate this, because you need maltose, which is a sugar, for making sweets or candies, or using it in some sweetening it in using it in some drinks, and using it maybe in the apple juice which you get across the counter. So we need the maltose. So we've taken starch and we've added amylase, and after some time we've kept it in a warm temperature, because you see the amylase works best at a certain temperature, and we have maltose, but we don't need the, so somehow we've got to remove this amylase. Now that's a pretty difficult possibility, because we need some sort of very special type of filters which are going to separate the maltose from the amylase. So some brilliant biologist came with the idea is, let's immobilize enzymes. Immobilize enzymes. Immobilize enzymes means that you would just get something and you would attach the enzymes to that. And then of course what you could do is, you could add it to the starch solution. So the starch which you had here, now we could easily do this, is we could add the starch here, and you could add these immobilized enzymes to this starch solution. And that would really solve the whole problem, because you see what would happen is that after some time the starch would be all finished, and what would we have? We would have maltose here, and then we could just remove these little immobilized enzymes which we have stuck to some sort of balls, or even we do it on the sides of this container, or we do it on metallic sheets. So this is called immobilized enzymes. And in immobilized enzymes, what we do is we can reuse these enzymes. So we can reuse these enzymes, and we don't need any sort of a very technically difficult filter by which we're going to separate the amylase. Now these balls of enzymes can be separated, can be removed, and then added to another solution of starch. The simple way for immobilizing enzymes is, you take an enzyme solution and you add alginate. Now alginate is some sort of thing which happens to gel very easily, and that alginate is then added to a solution of calcium chloride, and as soon as we add it to calcium chloride, this sodium alginate and the amylase just solidifies, and it forms these beads which you've done a number of times in paper 3. And what has actually happened is that these beads have the enzyme sticking to it. Please remember, enzyme is a very small molecule. So the enzymes are sticking to it. And now we can use these as we like. We can put them in starch, and we can then have them catalyze the reaction, and then we can reuse them again and again. So amylase and alginate put into calcium chloride solidifies, and you get these beads. And what is sticking on these beads is these enzymes, the amylase enzyme. So this is an example of immobilized enzymes.